Hello, my name is Sebastian Vuskovic. I'm a fourth year studying mechanical engineering at San Francisco State University. For the course Engineering 470 Biomechanics with Dr. Azadi, we were asked to create a presentation based on understanding and explaining research that we watched in class. And a quick little, I chose to take Engineering 470 with Dr. Azadi because I really enjoyed my mechanics of solids class I took, and I really wanted to apply those principles to the human body. The presentation I'll go over will just be some slides I chose from a research video we watched in class that I found interesting and I wanted to share. And these are all, none of this is from me. This is all from a video we watched in class. And let me show you where I got this from. Let me share my screen real quick. Cool. All right. So yeah, so none of this is my work. I got this contrasty of the Berker and Company and Dr. Garva, and the slides are adopted from the Berker's virtual event, which is going to be on the novel concepts in mechanical biology from individual cells to cancer tissues. So just repeat, this is just giving me slides I chose that I wanted to talk about, and this is all from them, all their work. Okay, so a quick little what we're going to do in this presentation is we had introductions, and then I'm going to have an introduction of Dr. Gavara, and then the rest of me 10 screenshots I took from her presentation, and there's going to be a thank you page. Okay, so this is going to be a quick introduction on Dr. Gavara and her research. So first, she started her schooling career to be become a physicist. And then she got a PhD in biophysics at the medical school of University of Barcelona. And then she took a postdoctoral position in the US and Germany. And then she became a lecturer of material sciences at Queen Mary University of London. And then she became a medical school University of Barcelona for her lab. So you could say that she has a wide range of skills and talents, but she's definitely focusing on a biomedical side of things. This is a picture of her right here. And the main focus right now she does in Barcelona is atomic force microscopy. And atomic force microscopy is a very high resolution type of scanning probe microscopy with demonstrated resolution on the order of fractions of a nanometer and more than 1,000 times better than optical diffraction limit. So very precise. Okay, so this is the first slide I chose. This is going to be what she does in AFM. This is just going through the measure of mechanical properties of living cells by indenting them. So this is the quick little process how she indents the cell. There's an indentation, cellular indentation, you got even further, a bead, and a parallel plate. I found this really interesting. Just I like how she just explained it off right. Like, this is what I do. Okay, and then she goes into the Psychoskeleton. Just did a previous project of tenserenity with the Psychoskeleton, so I was happy to see this. And the Psychoskeleton just provides the shape of the cell, and it basically allows the cell to change shape needed if the body needs it, like for development, body movement, wound healing, cancer, inflammation. So basically, the Psychoskeleton allows the shape of the cell to change to whatever need the needs the bodies need. And then we got the 3 cornerstones in cell mechanics. And quick little side note, cell mechanics is a subfield of biophysics that focus on the mechanical properties and behavior of living cells and how it relates to cell function. It encompasses an aspects of cell biophysics, biomechanics, and soft matter physics and cell biology. It encompasses a lot, but the 3 cornerstones is going to be a good atomic force microscopy setup, suitable contact mechanical model, and the optimal data analysis pipeline. Okay, so we're going to get a better, getting better data. And so Dr. Gavara, she stated, like, what does AFM truly measure? Just the things she asks her students in her lab. And as I explained, atomic force microscopy, what, like, you might think it measures force because atomic force microscopy, but no, it actually measures voltage or the voltage difference in the quadrant detector. So as we have seen this image right here, we got the signal readout of an electrical current, and it's right here. So yeah, so you just got to know that AFM doesn't measure force, it measures voltage, or the voltage difference in the quadrant. So I like that little clarification. Definitely got me. I was like, it measures force. And then we got deflection sensitivity. So deflection sensitivity is going to be as the displacement of an electrical beam at the target or screen of the cathode ray tube per unit of change in the deflection fluid. So this is a graph some of our students made with on their lab. So in the y-axis, we got the deflection sensitivity, and x, we got time. And as you can see, as the deflection goes down over time, it always goes back, it always bounces back a little bit. Next, we have depth sensing. It's a biology drives analysis. So depth sensors are a form of three-dimensional 3D rangefinder, which means they acquire multi-point distance information across a wide field of view. Standard distance sensing technology typically measures distances using one or more sensors with comparatively narrow fields of view. So think of it as a kind of like 3D viewing of the AFM. Then we go to spatially resolved cell mechanics. This is an idea, or like a way of thinking that one of her students thought up in her lab that she wanted to have, which I thought was really cool that she involved her students in this. So it's kind of, you start with analyzing the force right here, and then you gotta find the contact point, send the position of the contact point to create a map of the image. And then this was gonna basically define the contact point, like readily defined it. So this was a cool way of thinking of going one step to another. And then we got more data through better analysis. These are some ideas that I've already gone through in the video, but I just wanted to say them one more time. So we got through depth sensing, then we got viscosity with the classical protocol of FD curves. Then we got through topography or image style analysis. And then we got mechanical screening, mechanical profiling. Mechanical screening is report only the ones that have expected biological significance. And then mechanical profiling is identify measures better suited for classification. So mechanical profiling is definitely a little more naive, as you would say. It's kind of you choosing what you want. And mechanical screening is reporting the only ones that have expected significance. Two different fields of thought in there. The last one we're going to go over is the multiplex for machine learning classification. And Dr. Gavara basically said that mechanics of actin cortex and its linkage to the membrane is a lot more useful for learning this stuff, but not as useful as the mechanics of the nucleus, which has not given the best results. So basically, she's been focusing on the mechanics of actin cortex and its linkage to the membrane. Mechanics of nucleus, no go recently. All right, well, this has been it. Thank you for listening. If you have any questions, my email right here, my LinkedIn, if you want to get in contact with me. And again, none of this has been my work or ideas. This is from, if you follow this site, it takes you to this really cool virtual presentation where Dr. Gavara presents. I just wanted to find some slides that I enjoyed, and I want to go over them. Thank you for listening. I hope you have a good day.